The expression of eukaryotic genes can be regulated even after the transcription has been taken place. RNA interference is one such mechanism. Post-transcriptional regulation of gene expression means the regulation of gene expression after the completion of transcription. Here, mRNA is formed by transcription, but the translation process is regulated. Post-transcriptional mechanisms play a vital role in gene expression regulation of eukaryotes. One type of post-transcriptional regulation mechanism is RNA interference. In RNA interference or RNAi pathway, small non-coding RNA molecules were produced, and these RNA molecules will interfere with the normal expression of messenger RNA. That's why this mechanism is termed as RNA interference. Interference of gene expression by RNA molecules. Here you can see the structure of double-stranded interfering RNA molecule. They are also known by the names short interfering RNA, that is siRNA, and also as microRNA. Their length is approximately between 21 to 28 bp. Now let us analyze the different steps of RNA interference pathway. The first step is the formation of small double-stranded interfering RNA molecules itself. In this process, large double-stranded RNA molecules that were present in the cell were fragmented to produce small interfering double-stranded RNA molecules. This process is termed as dicing because the fragmentation process is carried out by an enzyme complex called a dicer enzyme. In this picture, you can see the fragmentation of large double-stranded RNA molecules to form small interfering double-stranded RNA molecules. During the second step, the small interfering RNAs make assemblage with the proteins to form ribonucleoprotein particles. Here you can see the small interfering RNA molecules associated with the proteins to form ribonucleoprotein particles. In the third step, one strand of the short interfering RNA present in the ribonucleoprotein complex get eliminated. The lost strand is the one which is oriented in 5' to 3' direction. The strand which is oriented in 5' to 3' direction get eliminated. The strand which is in the orientation of 3' to 5' remain associated with the protein. This structure with the single strand RNA with the protein is called RNA induced silencing complex, RISC. In the fourth step, the RISC targets a sequence in the messenger RNA that is complementary to the interfering RNA. The interfering RNA present in the RISC has sequences complementary to the mRNA. Here you can see the messenger RNA with the sequences complementary to the interfering RNA. So during the next step, the interfering RNA pairs with the messenger RNA. Notice the orientation of interfering RNA in RISC. It is oriented in the direction of 3' to 5'. This orientation is complementary to the mRNA which is in the 5' to 3' orientation. Here in this picture you can see the pairing of interfering RNA with the messenger RNA. The pairing has two outcomes. If perfectly base paired, the mRNA is cleaved and the mRNA is degraded. If the interfering RNA perfectly base pairs with the mRNA, the mRNA get cleaved and the mRNA is degraded. If the pairing between the interfering RNA and mRNA is imperfect, then the translation of the mRNA is arrested and the polypeptide synthesis from the mRNA is repressed. In either way, the expression of the gene get altered. So in summary, at first interfering RNA molecules were produced. Then the interfering RNA complexes with the proteins to form ribonucleoprotein particles. Then one strand of the interfering RNA gets eliminated, and this structure is called RNA induced silencing complex. RISC base pairs with the mRNA. If the base pairing is perfect, the mRNA is degraded, and if the pairing is imperfect, the translation of the mRNA get arrested. In both ways, the final expression of the mRNA gets altered.